Okay, video 3. This is actually really draining, and I am so out of energy right now, but we're gonna learn about fungi. Okay, so the diversity of fungi, learning objectives. Number 1, you should be able to, number 1, recognize the shared derived traits or synapomorphies that distinguish the fungi. And so first of all, we're gonna look at your tree, and because it's useful to know this tree. So you have your Luca, which is basically like the first animal ever, not animal, I'm sorry, it was not an animal, it was like most likely unicellular organism. Anyway, ancestor of like all living things. And then you have your bacteria, and then you have your archaea, and then you have your eukarya. And so far we've only looked at the breakdown of eukarya, and I know that it's gonna, this phylogenetic tree will be wrong once I put this all, like all the animal stuff and the fungi stuff here, but just keep in mind that all your nodes are supposed to like line up. It's actually kind of wrong right now too. But anyway, that's besides the point. So your eukarya, you're gonna have, I'm gonna draw this out first, and I'm pretty sure it'll be wrong, but being wrong is all part of the learning process. So that's how I'm gonna justify me getting this wrong. But you have plants. I know that you have plants of protists here. I mean, I could be wrong, but. And then you have like a third branch of like protists, but I won't put that there because that's annoying. And then you have, I think, this is fungi. And then this is broken up into coenoflagellates, and this is animals. I'm pretty sure that's how it goes. Plants, fungi, plants, fungi, coenoflagellates, animals. Okay, yeah. I think, yeah, I did a pretty okay job. Okay, recognize the shared derived traits, or that distinguish the fungi. So what makes the fungi different from an animal or kind of flagellate or a plant or a protist, right? And so let's look at some characteristics. So fungi are a clade. Their synapomorphies include absorptive, oh my god, I can't say that word. Okay, so fungi, they have I'm okay, I'll be okay. I'm just hella tired. Okay. Absorptive, was it heterotrophy? Yes, heterotrophy. And this means that they digest their food. They're not autotrophs, obviously, like they don't have like leaves or like they don't photosynthesize and things like that. And so they're gonna have to be heterotrophs, which means that they break down other organisms in order to get the carbon that they need to like generate things within their body. And so that's their source of like carbon, which is food. And absorptive just means that they're gonna be like we eat things and we put them in our bodies to digest and then like spit out waste and like use the other stuff. But in contrast, fungi secrete all this stuff, break it down outside of them, outside their bodies, and then absorb that carbon and like all the minerals and stuff. And then the second synapomorphy of fungi are chitin in cell walls. And although insects also have chitin, like you know that crunch that you feel when you when you step on an insect, that's the chitin. Chitin is actually a very structurally sound sugar molecule, and so that's nice to have. So, fungal mode of nutrition, absorptive heterotrophy. Fungi secrete enzymes into their immediate environment to break down nutrients and subsequently absorb them back into their cells. Yep, okay. So, chitin. Chitin reinforces cell walls. So, chitin is a modified polysaccharide molecule, haha, told you, polymer that forms a rigid component of fungal cell walls, adds structure and rigidity. Chitin itself is not unique to fungi. Like I said, chitin is also in insects, but its presence in cell walls is. So, whereas, insects might have chitin in their bodies, they don't necessarily have it in their cell walls. So don't memorize the structure of chitin, but can you recognize key features that you learned last semester? Okay, so I'm guessing that the blue numbers are supposed to say something. But basically, you have these two sugars that they're hexa. I don't remember exactly what the term was, but if I see it, I will understand it. It's like a ring of 6 carbons. No, 5 carbons, 1 oxygen molecule. Then you have a bond between the first and the 4th carbon molecule. Atom in the molecules. So 1 4 carbon bonds. So what do fungi look like? Most fungi are multicellular. The body of a multicellular fungus is called a mycelium. And so this is me. I am a human, but you don't call this human. You call this a body, right? And so if I was a fungus, you wouldn't call this fungus. You would call this entire body the mycelium. And that's it's the body of the fungus. The mycelium consists of many tube-like filaments called hyphae, which are used to absorb nutrients. And so they're like little arms that just branch out. And those branches are called hyphae. So multicellular fungi. The mycelia of many fungi live underground and can cover large areas. Mushrooms are spore-producing fruiting structures that grow above ground. The mycelial mass is often much larger than the mushroom. And so, one question that I would have for my professor is the mushroom also part of the mycelium? Which, it seems like a stupid question, but the PowerPoint words it in a way that it seems like it's not. So, here's the fruiting structure. / mushroom above ground. Also part of the mycelial mass / mycelium. Okay. Alright, some hyphae have dividing cell walls. Interesting. Okay. The hyphae of some mycelia are subdivided into smaller compartments by crossfolds, aka septa. Organelles can still move between compartments, however. That's such a weird concept. Okay, cool. Wait, let me look back at our learning objectives. So list characteristic features of the fungi as related to the general morphology, modes of nutrition. Know what yeasts are. Okay, so I guess we're supposed to also know. Okay, yeast. Unicellular fungi. Sometimes fungi are found to be single-celled. Single-celled fungi are called yeast. And so yeast refers to a lifestyle and not a clade. So some fungi species have both unicellular and multicellular life stages. Okay, yeast are a unicellular eukaryotic model organism of choice for many scientists. So yeast refers to a lifestyle, not a fungi. Some fungal species have both uni and multicellular life stages. And so, my question then is, if some fungi have both uni and multicellular life stages, then when that fungus is unicellular, is it called a yeast? I mean, if yeast is just basically like, it's a fungi, that's single-celled, then, interesting. So, how do fungi get their nutrients? Fungi differ in regards to where they absorb their nutrients. So, of course, I mean, it's absorptive heterotrophy, but there are certain ways that you can do that. So, some absorb nutrients from dead organic matter and others from living organisms. So, we'll consider 4 different examples, which is I'll get rid of this tree because we already know we're talking about fungi. So we have 4 different types of fungi when we're looking at like what are their appetites, what do they eat, and what's their relationship to the food that they eat. So you have saprobic fungi, you have parasitic fungi. I think you already know what that kind of means, parasitic, and then we have pathogenic. I think you probably also have a kind of a clue of what that means, but it's okay if you don't. Mutualists. Okay, so if we look at saprobic fungi obtain nutrients from non-living organic matter, e.g. dead plants. Some are called molds. Saprobes are major decomposers on Earth and critical to Earth's carbon recycling. Carbon recycling. Okay, I guess it just means that it like breaks it down, right? So this saprobic eats dead things. So when we think about like our common everyday mushroom, right? That's probably eating dead things. Probably grabbed the ground, probably grew on, when you see like mold on your apple, then that's also a kind of saprobic fungi. Parasitic fungi. Some fungi are parasites. For example, they invade plant leaves in order to absorb nutrients. Hyphae are well suited to absorbing nutrients from living plants. That's really kind of, wow, ouch. So the spores stick out of the little holes in the bottom of the leaves, and then like the root-like hyphae are all up in the cells of the inside of the leaf. I just got like goosebumps. That's kind of gross. Pathogenic fungi. Some fungi are pathogenic, meaning they cause diseases. A fungal infection is called a mycosis. Mycosis, like mycelium. Mycosis. So examples, athlete's foot in humans is caused by fungus. Most people with AIDS die of fungal diseases. Most of the important plant pathogens are fungi. Okay, all right. So fungi grow well in damp environments. What could make dry environments less suited for growth? I don't remember. Why? I think it's just harder for the fungi to like retain water, was it? And like stay in that environment. Why do fungi, okay, Google. Why do fungi prefer damp environments? Question mark. Okay, cool. All right. Why? Knows why. Here we go. Fungi live in moist environments because it is where they reproduce best. Due to their simple system, they need to grow in places that are moist and dumpy. This way, they will only have to rely on osmotic pressure, which is the pressure of a dissolved substance, to expand equally to all parts of the medium. It's also a perfect place for fungi which feed on extracellular digestion, which is all of them. They produce enzymes which digest their food. Okay, cool. I will accept that. So mutualists. Many fungi are mutualists, a relationship that benefits both partners. So 2 well-known examples. No. Okay, here we go. We have to know. So my core has associations between fungi and plant roots. Many vascular plants depend on these associations. Okay, so this is like the death cap mushroom, right? My core is If you eat a death cap mushroom, that's definitely gonna be true. Like death cap. And so basically what they do is they like latch onto the roots. They're like mycelial like body structure like kind of wraps around the roots of the tree. And so if you have like a wrapping around the root, then that provides more surface area for the water to get in. So that's how the tree benefits. But also the fungus itself is taking nutrients from the tree, and so it's kind of like mutualistic. Lichens, so associations between fungi and photosynthetic microorganisms. Okay. Lichens can partner with photosynthetic microorganisms that are not plants, typically green alga, most often observed on surfaces like rocks or tree branches. So sensitivity to air pollution. Lichens are sensitive to air pollution. This means that for humans, that makes them good indicators of pollution levels. Certain components such as sulfur dioxide appear quite toxic to lichens. Okay, a closer look at specific examples of fungi. Let's focus on Dicaria in more detail. So, within fungi, you have things that are not, so like etc., like other things, but like we're going to be looking at 2 particular examples of something called dikaryotes. And 2 examples are club fungi and sac fungi. Okay. So Ascomyota is sac. mycota. And Basidiomycota. Is club. Both groups can produce sexually, and they have a so-called dikaryon stage in their sexual life cycles. So fungi produce spores, which are single cells that are typically haploid, and undergo repeated rounds of mitosis to make a multicellular haploid structure. When a spore starts dividing, it is said to germinate, but make sure that you know that when they do start dividing, they're dividing in like mitosis, repeated rounds of mitosis. So you have a multicellular haploid structure. Fungal mating types. And interestingly, there is no morphological distinction between female and male fungi. We don't call them male or female. Scientists distinguish so-called mating types, either plus or minus. Mating types are genetically distinct, and only opposite mating types can be produced. So multicellular haploid structures that result from spore germination can be either plus or minus. In the dikarya, a hyphae of opposite mating types fuse. To produce cells that contain the unfused nuclei of both mating types. This is called the dikaryotic stage, a unique feature of dikarya. And so this might better be illustrated. Wait, is this for both Ascomycota and Mycota and the club fungi are most of, like, when you look at mushrooms, that's mostly club fungi. And Ascomycota, they have many multicellular members, but you can also look at lichens and be like, bruh, that's probably Ascomycota. Baker's yeast is also a single-celled member of this group. So I guess, oh no, wait. Lamau, okay, they have like similar kinds of, okay, they have similar life cycles. So let's look at the Ascomycota first. Right? So Ascomycota, when you have a life cycle thing, you basically look at some sort of life cycle. Like, obviously it's going to be like an actual cycle. Oops. Maybe I should draw it bigger. Okay. So you have this big part which is Lamau. It's not Lamau, it's just messed up. I'm going to draw this a little differently so that I can get more of a sense of, like, hey, I learned this. I'm going to draw it upside down. Okay. So a large part of the life cycle of Ascomycota is going to be haploid, which means you only have one set of chromosomes. The next part is going to be dikaryotic. And the whole meaning of that means that you have two sets of chromosomes. They're just not fused together. The nuclei aren't fused together. and then diploid, where they finally do fuse, right? These are the three major things that happen. And so let's start with the ascospores. And so when you have like little spore thingies, right, you have little spores. And then as we knew before this, those spores keep dividing in mitosis, repeated cycles of mitosis, and then that's called germinating. And so they become multicellular haploid structures. So then you get like hyphae. So then they just like add on, and then they like get bigger. And then you have like these little bulbs on top. And I mean like nuclei all over. But you have this bulb on top that's like a mating structure. And so if you have a positive mating structure here, let's say that like a negative mating structure comes in here. And so they come to these two bulbs like come together and fuse. And then what happens is that like after they fuse, right, they fuse together, and then they start creating more hyphae that way. And when they do fuse, that means that there's two different types of N. So this is no longer haploid when they, oh shit, wait, what? Lamel. Okay, cool. So right before they fuse, they're still haploid. So this thingy-mabobber should actually be right about here. So this is like the bulb thing. And this is the other bulb thing. Oop. That's a really messy drawing. But then they fuse and they create more hyphae. And so recognize that these two are fused, and so it's going to be dikaryotic. And this is called, this process, like the state that they're in right now, in the fused state, is called karyogamy. I mean, I lied. Plasmogamy. Which makes sense because their plasma is like fused. And then if you have so up here, you're going to have some sort of this, all of this is a fruiting structure. And here, down here, it's going to be, all this stuff is still going to be haploid. This is going to be diploid. I mean, dikaryotic. And so, after a while, like this just keeps spreading, down here. After a while, these things look like, they have like little pods that have, like, so like this, and that's like part of a bigger thing, like hyphae. But you have a positive and a negative and a positive and a negative. But they're not fused. The nuclei aren't fused. When they do fuse, however, they become diploid. Right. And then you undergo meiosis. Oh, when this is called, if that's plasmogamy, this is called karyogamy. Yes. fertilization. Wait, so does that mean that it's just... Oh, okay, yeah. So it becomes fertilized, diploid, and then it undergoes meiosis. And when everything undergoes meiosis, then it becomes haploid again. Right. There you go. And so with club fungi, it's pretty much the same thing. Focus on overall similarity to the Ascomyosis life cycle. Noteworthy dikaryotic part of the life cycle occurs. Okay, so I think that's it for fungi. My head is like spinning right now, but it's okay because I have one more PowerPoint to go, and that's animal diversity. Excellent. All right, I'll see you in the next video.